parasite of great concern. On behalf of our institute, we welcome you, Dr. Frey, and now over to you. Thank you very much for joining us.
Thank you very much, Rajnish, for inviting me. I will put that away so that we have the screen for the presentation. Yes, today's topic will be Echinococcus multilocularis, and I will give an introduction to this parasite because it's more of a concern in the northern hemisphere, and I'm actually not aware of any cases, any reported cases from India, but it's still a very, very interesting parasite, and I will first give you an introduction, general introduction, and then present two studies that I was involved with. One in Canada at the CFIA, where we did produce testing for the infectious stage of the parasite, which is the egg, depicted here. And another one will be the use of routine meat control to get an idea of the epidemiology of Echinococcus multilocularis in an area, and we did that with pigs. So I'm happy to start this presentation. Hope you're all okay, and I hope you can hear me. Yes, if not, please let Rajnish or me know, okay? Good. Why is this a why do I say it is a foodborne parasite of great concern? Well, it's not only my opinion, this is also the opinion of the WHO. They published in 2014 a top 10 list of the foodborne parasites of greatest global concern. And you can see number 1 is Taenia solium in pork, but then number 2 is Echinococcus granulosus, and number 3 already is Echinococcus multilocularis, and they say in fresh produce that this is really a big concern. Now, what are zoonotic taenid cestodes? So when we talk about taenid cestodes, we mean Echinococcus granulosus sensu lato, which causes cystic echinococcosis. This is depicted here, mainly in the liver, but also in the lungs quite frequently. And this Echinococcus granulosus is further divided into granulosus sensu stricto, which is G1 to 3, then Echinococcus ortlepi, and the Echinococcus canadensis cluster with the camel and pig strain and the northern cervid strains. Then we have further in the taenids, we have Echinococcus multilocularis. This is causing alveolar echinococcosis, also mainly in the liver. Over 90% of the lesions are in the liver. They look like this, more or less. As I mentioned, it is restricted to the northern hemisphere. And yeah, it targets primarily the liver. And then we have all the taenia, which are many, of course. And what we have here as main concerns for human medicine, again, is cysticercosis caused by Taenia solium and Taenia saginata, and also some other taenia. Here we have some nice Taenia saginata, cysticerci in beef. And today, or in this talk, I will mainly focus now on Echinococcus multilocularis. So the life cycle is, as it's usual for a cestode, it's quite complicated, and it involves multiple hosts. So we have the final hosts, which are canids. Sorry for that. Which are canids, dog, fox, coyote, and also the wolf. Wolves can be final hosts for Echinococcus multilocularis. Then they harbor the adult worm in their intestines. They shed eggs. Those eggs are infectious for the intermediate hosts, which are rodents, small rodents, and they will harbor the metacystode in their liver. So if they will develop this alveolar echinococcus, and then in turn they will be eaten again by the final hosts, and the cycle is closed. We do have final hosts, which can be important, but for the biomass of the parasite, they're not as important as the wild canids. We have, for instance, the dog, domestic dogs, or also domestic cats. But domestic cats, they don't really shed eggs of Echinococcus multilocularis, only very rarely. This is a puzzle. We really don't know why, but they don't seem to be very good final hosts for Echinococcus multilocularis. On the other hand, the dog is a very good host, and because the dog lives close to humans, they may represent actually a source of infection. Otherwise, humans, also dogs or pigs or other hosts, so-called aberrant hosts, because the cycle usually is a dead end, there doesn't go on. They get all infected by eggs, by the uptake of eggs. Now, human infections are rare, but they can be very severe. And in susceptible persons, the parasite is like a malignant tumor of the liver and can then spread to other organs. And this is why this parasite is ranked as high on the list of foodborne parasites of global concern, because really of the severity of the disease it causes in those patients. Now, this here is a very nice graph or figure I took from Thomson, 2017. This is the general life cycle of an Echinococcus, and we have the adult worm here in the intestine of the canids. Those worms are tapeworms, but they're very, very small. So they're just about 2 or 3 proglottids, 3 to 5 proglottids long, which makes them some millimeters. So they're really, really small. And the last proglottis is full of eggs. It gets detached and excreted. And those eggs here, they are already infectious. And they're very resistant in the environment. They harbor this oncosphere here, this little thingy with the hooks. And when that gets ingested by a suitable host, an intermediate host, the egg wall will break apart and the oncosphere is released. The oncosphere then penetrates the mucosa of the intestine and enters the circulation, and via the blood flow, it mainly arrives in the liver, and there it will grow and form this metacystode. And that can be a cystic metacystode or an alveolar echinococcus, depending on what we have here. Basically, what it forms are these little guys. These are protoscolices, so little heads. And when this metacystode then again is consumed or eaten by a predator, by a canid, those invaginated protoscolices, they will evaginate actively, and they go into the intestine, and with their hooks and those suction plates, they will then attach to the intestinal wall and grow again to maturity. And that takes about 4 weeks in the final host to happen. They don't live very long. They live 2 to 3 months in the final host, but there is no immunity. So one fox or a coyote or also a dog who was infected can be infected again. Okay. Now here I have some pictures taken at our institute here in Bern. This is an intestine of a definitive host, and all those little white thingies are actually adult worms, and I took a piece and magnified it a little bit. So all these white things here are the gravid proglottids of these Echinococcus multilocularis. And here we have a histology section of this piece of intestine. So you see the serosa and then the intestinal wall, and here the mucosa with the villi, and all these here are the worms. So it's really worm after worm after worm. And what is really interesting is that a definitive host can harbor thousands of worms without showing any clinical signs. So this is something you don't see if a coyote, a wolf, a fox, or a dog is infected, and that makes it very dangerous because they can really harbor a really high parasite load and contaminate the environment very heavily, and you don't see that in the animal. Okay, now this is in the natural intermediate host here. This is on the left, it's a rodent infected with infected with Echinococcus multilocularis. So, so we have this cyst-like structures filling the whole body cavity, the abdomen. And this rodent, of course, will then be very slow. It will be not as fast at running away from a predator anymore, and will be preyed upon more easily than a healthy one. And the other pictures here, what we see, this here is in a monkey. So monkeys can be intermediate host or aberrant host as well. This here is a dog. So the dog can not only be final host, but also intermediate host. And this here really is a pig with this large liver, and all these dots here are actually Echinococcus multilocularis lesions in the liver of a pig. And there are all other species that can be infected as well as intermediate hosts. Yeah, well, how can we get rid of that? Not at all. Okay. Well, seems we have to live with these green things.
Oh, okay, so maybe let me try if I can get rid of this, but
did I do that?
No, you didn't. I think somehow it is it's annotation.
Okay.
Let me try if I can do something.
Oh, great. Thank you. Okay, so I found some really nice images done by Katharina Kohn. I have them from the internet, but they're really nice. And they show alveolar echinococcosis in humans. So we have the stomach, the intestines, and here the liver. And in this liver, you see this cyst with this so-called brood capsules, and each of these little dots here would be a protoscolix. So if this liver were eaten, which is highly improbable, but let's think it through, of each of these protoscolices, a new adult tapeworm would be formed. Here it's the other one. So there are many, many brood capsules, and they are growing. So they can really, they can really infiltrate the liver and behave like a tumor.
Caroline, I think somehow you have to share your screen again, I think.
Do I have to screen again? Okay.
Yeah,
Sure.
we cannot see that.
Sure. Let me see. Yep.
Okay.
Okay.
Perfect.
Perfect. But you did see this slide, right? Or do I have to go on with this one? Okay, well, anyways, let's do it. So this is a human patient with the liver, and here the metacystode formed in the liver with these brood capsules and the protoscolices, and here as well the liver with the metacystode. And this metacystode really can grow and infiltrate the liver and also form like metastasis in other organs in a later stage of the disease. In humans, it takes 10 to 15 years after infection that the patient will really recognize that there is something wrong. And this happens mainly if the parasite is compressing either of the intestine or the gallbladder or something like that. And then usually the patient notices that something is wrong, and that will be like 10 or 15 years after infection. So there is a very long phase when this is just growing and we don't really notice that it's there. Okay, now on the next slide, this is of a paper by Bruno Gottstein and colleagues. They have characterized alveolar echinococcus in humans. And the A here would be immunocompetent and resistant. So in a CT scan, all you can see is this little white dot here. And if you resect that, it's a calcified, a dead lesion. So the parasite was here, but the body managed to kill it. And this is what you can find then, a lesion, but calcified and dead, and really nothing's going to happen here. Now, if you are immunocompetent but susceptible, then this parasite will grow. And this here is the lesion caused in a CT scan. So this would be the liver here. And this black hole really is the parasite. And on a resection, you see it like this cavernous, spongious material that is viable and that will grow on. And here on the right-hand side, we even have an immunocompromised host, who is highly, highly susceptible. So each of these lesions here in the liver would be a metacystode. And this is really spreading throughout throughout the liver first and then throughout the body also. And that's really, really scary. This picture here, or this figure, shows the current global distribution and prevalences of E. multilocularis in the main wild canid definitive hosts. And there you can see it's really the northern hemisphere where it's spread. And basically the darker a country, the more prevalent it is. And Switzerland is actually here somewhere. So we're in a zone of high endemicity. So it's really prevalent in Switzerland, but also in China, in Russia, in other countries, and in Northern America as well. What is scary and good to know as well is that the parasite is expanding in the geographical range. So it's really, yeah, it's gaining territory. And this graph here is now Switzerland, alveolar echinococcus in humans. In Switzerland, we have the human cases here. This is per 100,000 people, the cases. So first of all, you see that it's not so many, but what you can see here is an increase. And this increase is statistically significant. And it follows with a lag phase of about 10 to 15 years. It follows an increase in the fox population. So this here is the increase in the fox population. We had foxes, then the rabies was really taking its toll, and then rabies was eradicated and the foxes really, they proliferated. And then, so 10 years later, the alveolar echinococcus cases in humans started to increase as well. This is not only the case in Switzerland, you can also observe that in other endemic regions, that it's really that human alveolar echinococcus is increasing. And just to give you the numbers, so worldwide, we know of about 18,000 new cases per year. And the majority, the vast majority are in China, about 16,400. And in Europe, we're talking about 1,000. Hello. Okay. Now, if we come back to this top 10 list, we wanted, when I was working at the CFIA, we wanted to address this here. We wanted to address the risk of Echinococcus eggs in fresh produce and have a look if we could develop a method to detect them. And this is, this is how to detect E. multilocularis and other tenids in fruits and vegetables. This is published in Parasites and Vectors. It's open access, so if you want to read the paper, you can easily access it via PubMed, for instance. Now, what would be the perfect method to test produce? What should it be? What should it offer? Now, it should definitely detect and ideally also differentiate all the zoonotic tenids that can be around. It should be sensitive, so have a low detection limit. It should be specific, or if it's not so specific, then at least the positive results should be easily confirmable. Preferably, it's a quantitative method so that you know it's a high or a low contamination. It should be standardized and validated, of course, as any method you want to adapt. And it should have a potential to be integrated in routine surveillance programs. So what did we do? What did we have a look at? Basically the flow chart of how we test produce. Oh, we have other, hello. We have other little doodles here. Okay, we start with the contaminated produce. Then we need to remove the eggs. This is done by washing. We need to concentrate these eggs, extract the DNA, apply a PCR, and because tenids are, as I said, it's multilocularis, it's granulosus, and it's Taenia species, it needs to be a multiplex PCR that can find all of them. And then if we have positive results, they should be confirmed. So how did we do that? We spiked lettuce and strawberry in the lab with non-zoonotic Taenia eggs to reduce the risk for the people working with that. Then for the washing, we assessed 3 different wash buffers. The concentration of eggs was done by centrifugation, so the wash solution was centrifuged and that concentrated the eggs. The DNA extraction here as well, we assessed 2 different kits to extract DNA. And also for the PCR, there were different multiplex PCR were assessed for the performance with these produce washes. And the confirmation of the positive PCR results, that was done by sequencing. All right, now I walk you through this, through these experiments. First, we start with the PCR. We saw that the best results were observed with a PCR originally published by Traxel et al. In 2007. And what we did, that was a conventional PCR, and we changed it to a qPCR. And these are basically the results. So we have here Taenia species, Taenia pisciformis. We have in black Taenia saginata, in red Echinococcus canadensis, and in blue Echinococcus multilocularis. And these are the melting peaks and that are quite characteristic, and the amplification curves. And here it's again the performance of the PCR. So these are 10 to the 6 eggs of T pisciformis, 10 to the 5 to the 4 to the 3 to 2, and these are 10 eggs. So you can see that this standard curve is quite okay, that the PCR is really performing like we want it to do. Now, these are the different wash buffers that we assessed. So this is Alconox. Alconox is just a detergent you probably have in the lab, actually. Then glycine buffer, which is also used for this kind of produce washings, and sodium pyrophosphate. And here are lettuce and strawberries and the number of spiked eggs. And there you see that with sodium pyrophosphate, the mean CQ value was significantly higher than with Alconox or glycine buffer, and higher means less sensitive. Here now in the qPCR, you want low CQ values, means it's really sensitive. When they go higher, the sensitivity is less. For the glycine buffer, the same here with the 5 eggs, that was really higher than for the Alconox. And also in the strawberries, it missed some positive ones here as well. So what we used at the end was really this Alconox. We went forward with that one. And then for the DNA extraction method, we also had 2 kits to compare. And you see here the results. And again, this is the CQ value. So the lower, the better, meaning that the red curve here, the fast DNA spin kit, outperformed the DNA stool mini kit. So we used this DNA spin kit to go on. And the analytical sensitivity, of course, how low can we go in our spikes? This here is the number of eggs that we spiked. We didn't go lower than 5 because that is practically almost impossible. But the 5 were detected in 100% of the samples. So we know that our sensitivity is 5 eggs per sample or even lower, but we couldn't really test that. And the specificity. So what did we do for the specificity? We had different parasites that were spiked on produce like Giardia, Cyclospora, or Eimeria, and also tested some DNAs of other parasites like hookworm, Ascaris, Trichuris, Toxoplasma. And about 50 negative produce washes where nothing was spiked, but this is also very important to know if false positives can occur with negative washes. And they were all negative. However, this here is an example, these blue lines here, this is a negative. They can amplify very late in this PCR, but then the melt curve is clearly distinguishable. But this is something that can happen, and where we really will have to have a look at the melt peak to distinguish these false positives. Okay. Now, as a summary, this is more or less the summary. So we have the lettuce in the bags, in the stomacher bags. They get washed. These eggs will be recovered, DNA extracted, then the PCR, the melting curve analysis, and the positive ones you can even confirm by sequencing. So by melt temperature and then by sequencing. This is kind of the summary. And if you look at the conclusions between the perfect method and the method we established there, so the perfect method detects and differentiates all zoonotic tanids. Yes, it does. It detects them and they are distinguishable in the melt curve analysis or even in sequencing. It's sensitive. Yes, it's also very sensitive. We can go down to 5 or even fewer eggs per sample. Specific, yes, but not 100%. We can see late amplification. And so it's really recommended to sequence positive reactions when we would do that in a routine surveillance program. Quantitative, it is quantitative, but relative to your standard curve, standardized and validated, it's standardized at least, and it has the potential to be integrated in surveillance program. Yes, it has, but it would still need to be fully validated to do that. Okay, now that was the first story I wanted to share with you, and we're coming now to the second one, and this is a study that we've done in Switzerland and where we really profited from the routine meat inspection to gain information on environmental contamination with E. multilocularis eggs, and we did that in the pig. Also, this is published open access, so also here you really can, it's readily available via PubMed. Why did we use pigs? Now this is the cycle here, so we have the adults in the final host, we have the eggs that get excreted, the intermediate host, and this accidental host, and the pig is actually the most susceptible farm animal to E. multilocularis infection. And because it is really susceptible, it can act as a sentinel of environmental contamination with these eggs. What did we do? How did we design the study? Well, the 6 largest slaughterhouses in Switzerland participated in this study, meaning that we had access to about 85% of the annual pig production. It was designed for 1 year, and from every batch of pigs that displayed macroscopically altered livers, they took a random liver, a random sample, plus a control liver of a healthy batch of pigs. Now, batches of pigs means they lived on the same farm and arrived together at slaughter. And then we had like case farms and control farms. We did PCR and histology of these case and control livers, added a questionnaire interview with case and control farmers, and wanted to see the minimal prevalence of infection, the geographic distribution, and to identify factors that are associated with E. multilocularis infection in pigs. Okay, now the results of the PCR, what did we get? We got about 860 livers. Of these, 450 were submitted as case livers and 411 as control livers. We did PCRs on all of them, and we found of these 450, we only could confirm 200 as positive for E. multilocularis. The other 250 had lesions, but they were not due to E. multilocularis, or at least we could not pick them up in PCR. So we did histology as well on these lesions, and we could not find any anything that would say, yes, this is a T-need infection. No, they had like inflammation, fibrosis, things like that, but not echinococcus. 5 livers were positive for Taenia spp, so for different Taenias, not multilocularis, but others. And here we had these negatives. So the minimal prevalence in pigs was in finishing pigs, it was really 0.008%, so very low. And in breeding pigs, it was 0.1%, and this is a significant difference. So breeding pigs were significantly more infected than finishing pigs. How does this look like in the pig? It can be different presentations. Here we have all these white dots here are actually single lesions caused by E. multilocularis. And here we have a piece of liver with only one, but a bigger one. And this is a cross section of that same lesion. So the presentation can vary, but what we see, it's not like we had in the rodent or in the human or also in the dog. We don't have this growth. It's more really defined, a smaller lesion. And in the histology, we could also see this here. This structure is the laminated layer here as well. So this is very characteristic to Echinococcus infections. And this laminated layer was present in many of those lesions, not in all, but in many. And this is a clear indication of a multilocularis or an Echinococcus infection. But what we also see or don't see, and that's the important thing here, we don't see protoscolitis. So all those lesions were infertile, and actually they are dead. So this is really not growing anymore. We have the massive inflammation from the host around here with stars. This is inflammation of the host with host cells. And then also very characteristic for these Echinococcus lesions in the pig, we found these lymphoid follicles that the host was building around the lesions. And here with the plus, this even is a calcification, so it's already starting to really, well, the lesions are in the process of being removed from the liver. And that confirms that the pig actually is a very, very bad host for Echinococcus multilocularis. It kills the parasite, which is very interesting from an immunological point of view. So it's really able to kill that parasite, but we still can see the lesions in the liver. Now, if we have a map of Switzerland here, and then we see where we had our samples from. Here we have the pig density, so the darker, the more pigs. And this just shows us that, yes, we mainly had our samples from regions where there are pigs, which is kind of logical. And the red ones are the positive farms, the green ones are the negative farms, and the yellow dots are these other Taenia species. They were Taenia from the dog Taenia martis, for instance, from martens, or Taenia hydaticena we had. There is no clusters that we can identify, so it's really spread all over Switzerland. Now we also wanted to see the factors that are associated with infection in pigs, and we have risk factors. They are here in yellow. So the one that was really, really bad, really bad factor was foxes in the pig shed. Well, yes, if you have a fox running through your pigs and defecating there, that's really not good. Foxes on the farm as well was bad. Then we had here, this is a good one. This is showing us how many pig sheds a farm has, and the more it had, the less infection it had. So basically the bigger farms were less infected than small farms, and that can be because of hygienic measures or management measures that they would probably have taken. Here also other animals in the pig shed increased the risk of infection. Outdoor feeding increased the risk, and grass feeding increased the risk of infection with Echinococcus multilocularis. We also found some protective factors. like having a hygienic barrier. So before you enter the pig shed, you change your boots, you change the coat, and go in afterwards. That is definitely a protective factor. Also, deworming the sows 2 times per year was a protective factor, although the deworming will not have an effect on the Echinococcus in the liver, but it's probably just a sign of a good management. Rodent control was also a protective factor, and this one we were struggling a bit with. Dogs on the farm were protective factors, because I'm coming to that in the next few slides. Dogs can be infection risks for humans, and we actually anticipated that dogs on the farm would be a risk of infection, but no, they were actually a protective factor. And when we asked the farmers, well, what does your dog do on the farm? They all said, well, it guards the farm and it chases away other animals. So it will probably chase away those foxes, and the effect, the netto effect is actually a factor of protection. So. Good. If we compare that, this is a meta-analysis done by Conrads and colleagues with the risk factors for human alveolar echinococcosis. We see some that are really like very similar, like dog ownership is actually really a risk factors in humans, not with our pigs. We saw the other thing, but yeah. The grass also shows up with the humans. The foxes, here are the foxes that make a risk. And it goes on. We have again the dog ownership and the age over 20 years. Of course, the pigs don't get 20 years or older, but we had seen that the breeding pigs that are older than the finishing pigs are significantly more infected than the finishing pigs. So this is something that we can really relate our findings to findings from human medicine. All right. So the summary of this study is, or the takeaway message is that actually the routine meat control can be utilized to gain insight into multilocularis infection in pigs. We found a minimal prevalence all over the pig population of 0.09%, so low, but it was there. We could not identify any geographic clusters, so contamination with eggs is present all over Switzerland. And the risk factors that we found can be explained by the biology of the parasite. And we also found some protective factors that are due to hygiene and management measures. Yeah. These were the 2 studies I wanted to share with you today. The acknowledgements for the first study, definitely to the trappers and hunters who provided coyotes in Quebec, coyotes and wolves, and here the slaughterhouses and farmers in Switzerland who contributed to the second study, and the funding, the CFIA, the Western College of Veterinary Medicine in Saskatchewan, Canada, and in Switzerland, the Federal Office for Veterinary Science and Food Security Safety. And with that, I'm coming to an end, and I'm of course open for any questions. I think there are quite a few also in the chat. Thank you very much.
Thanks, Dr. Frey. So participants, you can ask and you can even write down in the chat box if you want. I think I saw there was a question in the chat box. Let me see if I can find it.
Sure.
So one of the question was by, so is there any seasonal variation? So I don't know. So whether that's question is about seasonal variation in intermediate or definitely host, but go ahead and yeah.
Yes. Well, in the intermediate host, I'm not really aware of seasonal changes, and we did not see any such thing in our pig study, so they came all over the year. But it is known in the fox, in the red fox, that mainly the young foxes are heavily infected, and mainly then when they have to leave the den. So that would be like late autumn, winter, beginning of winter. Then they are most infected.
Okay. And then there is another question. What ant helmintics we can use for this?
Oh, sure. Yes, absolutely. Well, in the definitive host, you would use praziquantel to kill the worms. It's really the drug of choice in the definitive host. In the intermediate host, you do it with benzimidazole, so with albendazole or mebendazole. And in humans, it's an albendazole treatment, and it's usually really a prolonged, so for many years that you have to take that. But in the final host, so dog and fox and coyote, it's one dose of praziquantel will kill the worms, and then it can reinfect itself, of course. So if you want to keep your dog free of these cestodes, you give them a dose of praziquantel every month, so all 4 to 5 weeks, and then you really break the cycle.
Let me see. What can be the prophylactic deworming schedule and drugs for humans?
The deworming schedule in humans with praziquantel.
Yeah. Yes, that's I think what the question.
Okay, you don't use praziquantel in humans for this infection. For humans, you really would need albendazole, and it's a daily dose, but well, I would have to look up the dose exactly. Not that I tell you something wrong, but
Okay.
it's a daily medication with albendazole.
So it's albendazole instead of
Yes.
praziquantel. Okay.
Yes, praziquantel is in the final host. Intermediate host is albendazole. Yeah.
Okay. So then one more question. Okay, what is the best treatment for this disease?
The best treatment.
Yeah.
Mm-hmm. The best treatment that we have right now is really, if you pick it up early enough and this lesion is still small, it would be a complete surgery. So if you can remove it by surgery completely, then you would still give some albendazole just to prevent spreading throughout the body. But then you have a really good chance that you get rid of it. If the lesion is too big to be removed, then all you can do really is the albendazole, and the albendazole will not kill the parasite in your liver, but it will at least stop the growth.
Yeah, it was a good question by Archana. Okay,
Yeah.
the next question is, pigs are considered to be as sentinels. Why this despite their ability of killing the cyst? Why not surveilling on carnivores or definitive hosts?
Yes. Yeah, this is also a very good question. Why did we use the pigs? Well, basically, if you want to do a study on the carnivores, that's great. That's absolutely a good idea. But you will have to look that you get the samples. We use the pigs because when they are slaughtered, each and every pig is going into meat inspection. So the inspectors there, they look at those liver. They will see those lesions. And even if it kills even if the pig kills the parasite, the lesions are still there. And that is just a very economic way, I think, to get an idea of the situation in an area, because these pigs are controlled anyways. You can just kind of profit from that routine inspection to get more information on something that is otherwise really complicated or tedious to get the samples. That's why we use the pigs, and the pig is, as I mentioned, really the farm animal that's most susceptible to this infection, although it can really kill the parasite, which is really interesting, I think.
Okay. Any other question? Okay, any other question? Okay, there is one more. What do you mean by meat control? Do you mean hygienic meat or some other concept? Which anthelmintic drug is highly sensitive and which one is resistant in your part of world? Okay.
Okay, the meat control, that really is in the slaughterhouse when the animals get slaughtered.
Okay.
The organs will be of each animal will be presented to a veterinarian. This is just a routine meat inspection in Switzerland. And the veterinarians in the slaughterhouses, they are trained to look for pathological findings. And they also have a look at the meat, at the body. So it's the organs and the meat, and they really inspect it visually and can take samples if they think it's appropriate of each and every animal slaughtered in these slaughterhouses. I don't know how the situation is in India, if this is the same, but in the larger slaughterhouses in Switzerland, this is really the protocol that has to be followed. So each individual animal will be controlled after slaughter.
Yeah. So there is another question, I think, which I will try to answer if I can. So whether in India the infestation is there, then in which area? It's really a good question. So if we talk about India, so we do have Echinococcus granulosus, all right? So which is another member of this family, Echinococcus. And this is prevalent in some parts of India. And if I talk about in northern India, there was a study which was done in humans in northern India, and the different strains which were found, they were G1, G3, G5, and G6. So we do have cases, all right, but however, the life cycle is same, but the definitive hosts and as well as the intermediate hosts, they are different, right? Yeah. Do you want to say anything, Caroline, about this? Would like to add something?
With Echinococcus granulosus, you have not rodents as intermediate host, but really farm animals like cattle, like pigs, camels, sheep, goats, and also again humans can be infected, right? And for definitive hosts, it's mainly dogs and wolves, less foxes. So a little bit bigger. Everything is bigger there. Even the cysts are bigger.
Yes, yeah, agree. So that was a good question by, I think, Dr. Javeed Khan. And then there are a few more. I will quickly take some of those. What preventive measures we can take to prevent this? That's a good, and I would like to add into it, Caroline, you can add my question also to it. You talked about fruits and berries, they can also get contaminated. So when we buy something, so is a normal washing can remove those eggs from the skin of these vegetables or fruits? So what's your take on this, as well as about the preventive measures we can take?
Yeah, preventive measures, exactly. So with food, vegetables and fruit, I would really wash them, if possible, with a little bit of soap, because that will remove the eggs better. But if not, thorough washing will probably be good enough. And if you can, well, these eggs, they will die when you heat your food. So everything that's heated is safe. Okay. But they survive freezing for quite a long time. So this is not really an option. And the other preventive measures are really, like, if you live with dogs, that you deworm them regularly with praziquantel all 4 to 5 weeks. And if you are in contact with wild canids, that then you're really extra careful that you wear gloves and if possible a mask when handling those, for instance, to get the fur or whatever. So these are kind of the measures that you can do or that you can take. And also if you're gardening, if you have a vegetable garden, you should wash your hands afterwards and try not to pick the things directly and put it in the mouth. So really collect them, wash them, and wash your hands.
Yeah. Even while you are doing gardening, you can wear gloves. So that can also be make you.
Yes, having a kitchen garden was actually one of the risk factors. Yes. For humans.
Okay, so I think the last two questions. So one is which other intermediate hosts would replace a sentinel consideration of pigs, taking example of communities who don't rear or slaughter pigs.
For multilocularis, it's difficult because multilocularis does not cause any problems in ruminants. So we cannot really replace the pig by ruminants. It's a good question. Birds also, they don't, so poultry you cannot use. Then you would probably really have to go for the rodents. But that's, rodent surveillance. But of course, that's more complicated to get samples probably, yes.
I agree. And one last question is, are there countries free from this disease? If yes, what strategies they have adopted?
Yes, there are many countries that are free from E. multilocularis, the whole southern hemisphere, but they usually have E. granulosus. So is there a country that's free from both? Probably Greenland and probably the Antarctic. But other than that, it's really distributed very, very widely if we look at granulosus and multilocularis. Yeah.
Okay, I think there are no other questions, and so I would like to thank Dr. Caroline Frey on behalf of my institute. So she accepted our invitation and spare her time and put her efforts to give such a nice, wonderful, interesting talk for all of us. And we hope participants have enjoyed.
Thank you very much.
And I would also like to thank Dr. Guman, who is the Dean and PI of this project under which we are running this webinar series, as well as Dr. Jill and Dr. Cooper. And at the end, I will also like to thank on behalf of School of Public Health, our Honorable Vice Chancellor, Dr. Inderjit Singh, for supporting us for organizing such events. So thank you very much. And at the end, all the participants, thank you very much for joining us, for showing up. And I am really happy and glad for your active involvement, asking questions and having discussion with the speakers, not in this session, but also in the previous one. So, and I hope you will enjoy the another webinars which will be in the coming weeks, as well as actively participate in those two. And with these words, I will again thank Dr. Caroline Frey. Thank you very much. Thanks a lot. See you soon. Bye. Thank you. Bye. Thank you. Okay. So I think we can see you next week.